Gene therapy. Historically, the discovery of recombinant DNA technology in 1970 provided the tools to efficiently develop gene therapy. Scientists use these techniques to readily manipulate viral genomes, isolate genes, identify mutation involved in human disease, characterize and regulate gene expression, and engineer viral vectors and non-virus vectors. The gene therapy technique was first developed in 1972 but has so far limited success in treating human disease. So, what is the gene therapy? Gene therapy is when DNA is introduced into a patient to treat genetic disease. The new DNA usually contains function gene to correct the effect of a disease-causing mutation. Gene therapy may be a promising treatment option for some genetic diseases including muscular dystrophy and cystic fibrosis. This technique may allow doctors to treat disorder by inserting gene into a patient's cell instead of using a drug or surgery. The DNA is carefully selected to correct the effect of mutated gene that is causing disease by replacing the mutated gene that causes disease with healthy copy of the gene. Inactivation the mutant gene that is function improbably, introducing a new gene into the body to help fight a disease.
How does gene therapy work? In cancer, some cells become diseased because certain genes have been turned off. By using gene therapy can make two pathways. The first one turn off the mutated genes that cause the disease, and the second one is turned on the healthy genes that can inhibit the disease. The genes delivered into cells by a carrier called vector.
Gene augmentation therapy. This is used to treat a disease caused by mutations that stop a gene from producing a functional product. This therapy adds DNA containing functional version of the lost gene back into the cell. The new gene produced functional product at sufficient level to replace the protein that was originally missing. Gene inhibition therapy, the second technique. This technique is suitable for treatment of infection disease, cancer, and inherited disease caused by inappropriate gene activity. The aim to introduce a gene whose product either inhibition the expression of another gene or interfere with the product of another gene. This basis of the therapy is to eliminate the activity of the gene that encourages the growth of disease-related cell. Killing of a specific cell is the last technique we will discuss. It is suitable for disease such as cancer, which is treated by destroying certain group of cells. The aim to insert DNA into diseased cell which cause cell die. This can occur by two ways. First, insert DNA contains suicides gene that produce the highly toxic product which kills the diseased cell, or inserted DNA causes expression of a protein that marks the cell so that the diseased cells are attacked by the body's natural immune system.
Challenges of gene therapy: delivering the gene to the right place and switching it on. Making sure that the new gene doesn't disrupt the function of other genes. Gene therapy is very effective but may also be very expensive. Gene therapy is not completely safe as it can be followed by reactions such as unwanted immune system reactions as inflammation, targeting the wrong cells, infection caused by the virus, and finally possibility of causing a tumor if the new gene gets inserted in the wrong spot in your DNA. There is a debate concerning the moral and ethical issue involved with gene therapy. Through the use of genetic engineering technique, DNA can be sequenced, analyzed, and altered. This manipulation of DNA can be seen in either a positive or negative light, depending on the individual involved. Some individuals support gene therapy and some are not.
Types of gene therapy. Somatic cell gene therapy. Therapeutics genes transferred into somatic cell will not be inherited later generation. At present, all research are directed to correct genetic defects in somatic cell. For example, introduction of gene into bone, blood, skin cell. Somatic cell is non-productive. Therapeutic germ gene therapy. Therapeutic gene transferred into germ cell. It's heritable and based on later generation. For safety, ethical, technical reasons, it's not being attempted at present. For example, genes introduced into eggs and sperm. Approaches technique in genotherapy. In vivo genotherapy, copies of therapeutic gene are inserted into viral DNA liposome or in form of plasmid DNA. Second, genetically altered DNA is inserted into patient's body by cell-specific direct tissue injection. Third, inside the body, the inserted DNA is incorporated into cell of the specific tissue. It was injected into this cell, now encode and produce the needed protein encoded by the inserted gene. In ex vivo gene therapy, copy of therapeutic gene, target cell removed from the patient, a cell grown in the culture and the culture cell are infected with genetically altered virus. Cells are reintroduced in the body. Inside the body, the genetically altered cell produces the desired protein encoded by the therapeutic DNA. Types of gene therapy vectors. Non-viral vectors, naked DNA, liposome DNA, polymer DNA complex, liposomes polymer DNA, and viral vectors. I have two types, DNA viruses and RNA viruses. DNA viruses include adenoviruses, herpes simplex viruses. RNA include retrovirus. Viral vectors include retrovirus, lentavirus, adeno-associated virus, adenovirus, herpes simplex virus. I will talk about type and advantage for each one. Retrovirus type integrate with the host chromatin, and its advantage effective for long period of time with low immune response in host. Lentavirus integrates with the host chromatin. The advantage transect prophylactic and non-prophylating hosting. Adeno-associated virus type Ether advantage very good length of expression and efficient transfection in vivo. Adenovirus extra chromosomal DNA and advantage high efficient transfection in vivo and ex vivo. Herpes simplex virus the type extra chromosomal DNA very good length of expression especially in vivo. Safe use in immunocompressed patient.
I will talk about using of gene therapy. Gene therapy technique have the potential to provide alternative treatment for those with infertility. Recently, successful experimentation on mice has proven that fertility can be restored by using the gene therapy method. Second use of gene therapy technique is gene doping. Athletes might depend on gene therapy technique to improve their performance. In addition to genetic engineering could be used to change the physical appearance, metabolism, and improve physical abilities and the mental faculties such as memories and intelligence.
Disadvantages of gene therapy. Gene therapy, though, provides means of treating diseases, but still this therapy is not very common because it has some limitations. Maybe the therapeutic genes which are inserted in the body are not accepted by the immune system, and the viral vectors are not useful because they can trigger their pathogenic features in the body. The most common gene therapy risks: the unwanted immune system reaction, targeting the wrong cells, infection caused by the virus, and the possibility of causing a tumor.